Good morning and welcome to the Genenta Science Expert Perspectives on Immuno, Gene, and Cell Therapy for the treatment of solid tumors. At this time, all attendees are in a listen-only mode. A question and answer session will follow the discussion. If you'd like to submit a question, you may do so by using the Q&A text box at the bottom of the webcast player or by emailing your questions to questions@lifesciadvisors.com. As a reminder, this call is being recorded and a replay will be made available on the Genenta Science website following the conclusion of the event. I'd now like to turn the call over to Luigi Padacchi, co-founder and chief executive officer of Genenta Science. Please go ahead, Luigi.
Thank you, Tara, and thank you all. So I see here connected many institutional investors, sell-side analysts, and current investor of Genenta. So I'm very excited. More than 100 person registered for this event, so I'm very glad for that. As Tara said, I'm the CEO and co-founder of Genenta Science, and at the Genenta, we have decided to support this event since we are working hard to reprogram the highly immunosuppressive tumor microenvironment in solid tumors. We are developing a cell gene therapy strategy to deliver within the tumor microenvironment potent anti-tumor payload able to reactivate the immune response, this avoiding toxicity. Our product, named Temphron, different from a CAR T, is tumor agnostic and antigen agnostic. So we are here to learn more from this fireside chat. Since we are a listed company, we need to show you the forward-looking statement. And let me introduce the panelists for today. Professor Luigi Naldini, that is the director of the Raffaele Telethon Institute for Gene Therapy and co-founder of Genenta. Professor Reardon, clinical director at the Center for Neuro-Oncology at the Dana-Farber Cancer Institute. And one of our advisors, but more, one of my best friends is Loncar, one of the great voice of the sector that will be the moderator for today. So I leave the Zoom floor to Brad.
Thanks a lot, Luigi. It's great to have the chance to moderate this morning, and truly a global event. I'm in Kansas City this morning. We've got Dr. Naldini in Italy, of course, and Dr. Reardon in Boston. And I know we have listeners from all over the world, so I'm excited to have a great discussion. And we also have Carlo Russo with us, who is Genenta's chief medical officer, in case any Genenta-specific questions come up that he might be able to help us with. So we have a lot to talk about today. I'm going to jump right into this. Let's start with you, Dr. Reardon. Let's set the stage for glioblastoma. Can you describe the current treatment landscape and unmet need for patients with glioblastoma?
Yes. Hello, everybody. Glioblastoma is the most common type of cancer to arise in the brain in adult patients. Brain cancer represents a disproportionate amount of mortality and death amongst oncology patients today. It only accounts for 1.4% of all cancers, but it has now become the number 10 cause of cancer-related death in both men and women, and it's the number 1 cause of cancer-related death in children. There are approximately 10 to 15,000 cases of glioblastoma diagnosed in Europe, as well as a similar number in the United States each year. Our current standard of care was defined back in 2005, 17 years ago, and includes maximum safe surgical resection, followed by radiation therapy, which is typically directed to the tumor over 6 weeks, and usually given in conjunction with an orally administered chemotherapeutic agent. After the 6 weeks of radiation is done, patients then go on and receive approximately 6 months of oral chemotherapy. With that standard of care, unfortunately, although tumor can be temporarily controlled, the tumor activates and recurs, and patients ultimately die of the disease, typically within a year to 18 months. There have been several large clinical research efforts done trying to define a new standard of care to improve outcome. And unfortunately, despite the efforts, these have not been successful. and a variety of different agents have not proven the ability to improve survival for patients with glioblastoma.
Well, Dr. Reardon, what has been the barriers to success for those emerging treatments? What have been the difficulties and why haven't they succeeded in the past?
There are a variety of them, Brad, that have made this a very difficult and challenging tumor to try to treat. First and foremost is the fact that this is a tumor by definition that arises in the brain, and about 90 to 95% of the cancer therapeutics that are currently used for the treatment of colon cancer and lung cancer and breast cancer, they don't get into the brain effectively. Mother Nature designed us with a structure called the blood-brain barrier that is normally in place to help protect the brain, an extra level of protection against anything that could be harmful that makes its way into the bloodstream. But unfortunately, that blood-brain barrier works against us for the treatment of brain cancer because most of the therapeutics that are able to be helpful and successful for other cancers simply don't get into the brain effectively. They can't penetrate through that barrier. So our patients have an additional and unique challenge that's distinct from other cancers, where we have to have therapies that can successfully get into the tumor in the first place. In addition, what I tell my patients is we have treatments that can help you, but nobody has to find a treatment that works durably. The treatments we have, the tumor has the ability, a remarkable ability to adapt and become resistant, whether it be to chemotherapy, even radiation therapy, biologic targeted therapies, anti-angiogenic therapies, many of the treatment forms that have been helpful for many different types of cancers. Unfortunately, with glioblastoma, although they may work for a brief period of time, the tumor has a remarkable adaptive capability to develop resistance. And that's a second important challenge that we need to overcome to improve outcome for this disease.
Okay, and are there any emerging treatments or technologies that you think are particularly promising that maybe excite you today?
Yes, I think there are actually. As many on the call may be aware, immunotherapy has transformed how we treat many cancers today. The development of checkpoint blocking agents, the ability of vaccines, cellular therapies have changed the landscape of how many different cancers are now treated. I think in the United States, there's, last I tabulated, 62 different cancer approved indications for immunotherapy treatments. And these immunotherapy treatments are now replacing chemotherapy, radiation, the historical treatments we've used, which can be quite toxic for patients and have a lot of side effects that can impair quality of life and function. Immunotherapy for brain cancer, unfortunately, the initial trials, particularly with checkpoint blockade, PD-1 blocking agents, have not been successful. But there is a tremendous amount of research that's ongoing now to better understand why tumors like glioblastoma are resistant to some of these immunotherapy agents. And we know from that research, one of the important pivotal factors relates to the tumor microenvironment, where the tumor generates a milieu around the tumor that specifically is designed to block and inactivate our body's natural defenses, the immune system's ability to attack the tumor successfully. So I'm hopeful and optimistic that although the first generation studies have not yielded the positive results we'd hoped for, that with persistent research and ongoing efforts, we'll better understand the biology of these tumors, particularly that aspect that they're exploiting strategies and mechanisms to block the immune system. And with that knowledge, we'll be able to develop therapies, I think like Temphron, for example, that can overcome some of these protective mechanisms glioblastoma tumors are utilizing, and then allow the immune system to be able to successfully attack the tumor.
Excellent. Well, I think that's a great point to bring Dr. Naldini into the discussion. Dr. Naldini, good morning, or I guess good afternoon where you are today.
Good morning to you and to all the who are listening to us.
So Dr. Naldini, you've led the development and application of lentiviral vectors for gene transfer in rare diseases. What's the rationale for applying those same techniques in cancer?
So Brad, what we really have been developing is stem cell-based gene therapy. So modifying hematopoietic progenitor and stem cell of a patient with a vector that would replace a malfunctioning gene. Now, what happened when these cells are reinfused and engrafted in the patient is that their progeny will distribute throughout the tissue and also migrate into a remote area. This providing functional correction of the diseases. And we found that this can be very efficient and safe and also long-lasting to the patient. Now, some of the diseases that we have been treating are based on a defective lysosomal enzymes, and the corrected cell which migrate into the tissue also release some of the enzyme, which becomes available to the neighboring cell, also helping in this protective effect against the symptoms. So from that observation, I think we started to thinking that instead of working to replace a malfunctioning gene, we could use the same strategy to distribute and target biotherapeutics to remote area. For instance, in a tumor in the brain, like as Dr. Reardon said, maybe protected by the blood-brain barrier. But in this case, the gene-modified cell would actually actively migrate through that barrier, reaching, for instance, the tumor and delivering a biotherapeutic. So this concept of essentially gene-based delivery of biotherapeutics through smart carriers, which are cell capable of actively migrating to tissues and in particular to disease site, is the key of what we have been developing.
Well, Dr. Reardon, let me ask you, what Dr. Naldini just described is the cutting edge of science. You know, that's a lot different, of course, than a checkpoint that's infused or, you know, certainly a pill in a bottle. So your field, neuro-oncologists, haven't engaged much with gene therapies and stem cell-based interventions. So before we delve into detail, why is that? And do you think that the lack of exposure could be a potential obstacle to wide acceptance among clinicians if clinical evidence supporting this type of approach is forthcoming?
Well, I think there may be two reasons contributing to some of the sentiments in neuro-oncology about cellular therapies and stem cell therapies that, and they relate to the challenges basically that I discussed earlier. First of all, we have to get, these cells have to be able to traffic effectively into the brain, into the tumor, in order to be able to have an effect. CAR T cells, for example, we know have been remarkable against various leukemias, multiple myeloma. They're injected directly into the bloodstream, and they can effectively seek out and destroy the target expressing cells that are systemically in the body and available. For that type of an approach or other cellular approach, stem cell cellular approaches, for our patients, for brain cancer patients, these cells have to get into the brain. So there has to be a strategy or a mechanism whereby they will biologically be led or preferentially trafficked to the tumor in the brain, or directly administered into the tumor in the brain. So I think number one issue has again been the issue related to getting into the tumor in the brain. That's the additional challenge we face in brain cancer. And secondly, is the factor of time. Many of these cellular therapies can take 2, 3, 4 months to generate the product from the patient in order to administer to them. And unfortunately, for a disease like glioblastoma, patients may not have that amount of time in order to be able to, for the therapy to be generated and be able to be effectively administered. So we need something that can be generated relatively quickly to be able to administer to the patient in a timely manner, and then be able to successfully get into the brain.
And in terms of potential uptake, if a therapy succeeds in the clinic, just based off of this being a little different than, maybe what current practice is, do you see that as a potential barrier to entry, or does, efficacy and safety trump everything?
Well, obviously the bottom line, of course, is always going to be safety for our patients and improvement in survival and outcome. But it would be a very positive, exciting development to be able to demonstrate, first and foremost, that the drug, the treatment, the cells are getting into the tumor and having an effect on their intended target. For glioblastoma, that's not easy to demonstrate. It's always been a challenge for our field. And we've had many unsuccessful trials over the years. And at the end of the trials, we don't know why the drug or the treatment didn't work. Was it because it just didn't do what it was intended to do successfully and effectively? Or in many cases, I suspect it just didn't get into the tumor effectively in the first place. It may very well have been a good drug with the ability to do what it was intended to do, but just not able to get into the tumor effectively. So for our field, if we can certainly demonstrate safety and an improvement in survival, control of the tumor, obviously those are very, very critical steps. But also, I think it'd be very important to have some proof of principle and biologic validation that the drug, the treatment, is truly being able to get into the tumor and have an impact on its intended target. That would be very exciting for me and other clinicians and investigators in our field.
All right, and Dr. Naldini, you talked about your work at a very high level, but let's delve into Genenta's approach specifically. So the idea here is to create gene-modified monocytes for tumor-restricted expression of interferon, and the lead program is called Temphron. Can you describe in more detail what Temphron is and how it works?
Sure. Let me take this through some steps. So first of all, do we have a biotherapeutic that we want to deliver to the tumor? Yes. You mentioned interferon. That's what we think is a very powerful pleiotropic activator of immunity. We know it can work against tumor. It can also work against viral infection. But if you administer it systemically, you have a toxicity which prevents effective and safe administration. On the other hand, if we could deliver interferon selectively to the tumor site, sparing the periphery, we could have all the benefit of its activity without the side effect of systemic exposure. Second, what is our delivery vehicle? That's something, as I mentioned before, is a cell, is a normal body cell, which will go naturally into the tumor. And what we are using here is a leukocyte, so it's a white blood cell, is a monocyte, which is naturally recruited into the tumor. And unfortunately, this is usually done for the tumor to grow. So the tumor needs those type of infiltrating monocyte to foster vessel growth and its own growth. And there is a requirement for that. So this is not an option for the tumor to do without that. Now our strategy, we modify that cell to carry within the tumor the interferon gene, like a Trojan horse in principle. The tumor needs that cell, but once the cell reaches the tumor, it will locally deliver an anti-tumoral payload. So that's the key aspect. And because it's only the tumor which is recruiting in a normal adult this type of monocyte, you will have selective expression and delivery of interferon at the tumor site. And we do this through a lentiviral vector, which is a gene transfer vehicle which we introduced into the hematopoietic progenitors, which are, of course, the source of the monocyte, which are continuously generated into the bloodstream. But the vector is designed so that it will only activate expression within the tumor, essentially responding to the differentiation program which the monocyte undergo when they reach the tumor. And that is the key design of the treatment.
And I'm wondering, so you talked about interferon and how to improve on that for this disease, but with this technology, you could have chose to test it in anything. I'm wondering why you decided to choose newly diagnosed glioblastoma patients for the initial testing, and if there's any preclinical work that gave you the confidence to proceed into the clinic with this treatment and with this specific cancer.
Oh, you're perfectly right, Brad. In fact, we did test this treatment into preclinical model, into mouse model, of course, of tumors with different tumor type, and the principle are general. And in fact, the treatment had efficacy on a variety of the tumor. When we came to the choice of the first human testing, of course, there's a number of criteria. The first one is the high unmet need. So tumors for which there are very few options available. And as Dr. Reardon, I think, well explained to us, unfortunately, glioblastoma is one of those. Plus, glioblastoma is also well known to be highly recruiting this type of immunosuppressive monocyte, and so it is lend itself pretty well to our strategy, and it's also well known to have a strong immunosuppressive microenvironment. Plus, it's hard to reach, and we had this strategy we could use actively migrating cells. So overall, I think it provided us the appropriate risk-benefit framework for a first-in-human testing of a new strategy. And finally, I would say the patient who are diagnosed with glioblastoma, despite such a dramatic diagnosis, they usually are very healthy, on the other hand, body-wise. And so the possibility to engineer hematopoiesis for this targeted delivery is well applicable into this setting. And finally, sorry, of course, the key point is, do we have preclinical evidence? Of course, we had to build serious preclinical evidence of the safety and potential efficacy of the strategy in glioblastoma mouse models, which we did indeed. We had reported already and some studies in glioblastoma model. There is also a paper coming out soon, actually, with more detailed study, which were part of the material used to get approval for the trial.
And Dr. Naldini, you mentioned a key word, risk benefit. Especially in the early stages of testing anything, you're looking very carefully to make sure that a potential therapy is safe. There have been some recent safety concerns with lentiviral gene therapies. There was in the news a patient who developed MDS in a cerebral adrenoleukodystrophy trial that the investigating companies determined was likely mediated by the lentiviral vector. Having treated hundreds of rare diseases patients with lentiviral therapies, what was your analysis of that? And should we be concerned about any theoretical safety issues with Temphron?
Well, for some reason, we tend, of course, in the news to look at the adverse effect rather than the benefits. We have solid clinical experience of lentiviral vector gene therapy, both in hematopoietic stem cell as well as in T cell. But just staying with hematopoietic stem cell, as you said, we have treated more than 120 patients. Probably worldwide, we reach about 400. The outcome in all of these patients, I think, extremely positive, not only in terms of safety, but also in efficacy. And we have a strong understanding of that. We have molecular monitoring of the hematopoiesis into this patient, which show very benign behavior of the vector, except, as you said, for this specific trial, which reported recently this adverse effect in one and maybe a second patient as well. Now, this is something that to a certain extent could also be predicted because of the specific design of that vector, which increased the risk of genotoxicity. So I think this is something that should not worry us more than what is the usual careful approach to any medicine, which has, of course, potential for benefit and for adverse effect. In the specific scenario of cancer treatment, I think the type of vector design that we are using in this setting is certainly really making the concern for this genotoxicity almost minimal to none, honestly.
All right, and I'll go back to Dr. Reardon to maybe get your reaction to that. From what you've heard about Temphron, what outcome from a phase 1/2A study would persuade you from a safety and efficacy perspective that this is a serious option given the risk profile and current options out there for patients with this disease?
Well, I'm very excited about this particular therapy and approach because I feel it gets right to the heart of the problem we face, the challenge we face for immunotherapy in this disease, and that is the myeloid component of the tumor. If you look at a photograph of a glioblastoma tumor, and you identify all of the tumor cells in that photograph. If you look for immune cells that are in the tumor, T cells and other immune effector cells, out of 1,000 glioblastoma tumor cells, we may see 1 or 2 T cells. They're very few. In contrast, we will see hundreds of myeloid cells. And these myeloid cells are attracted into the tumor by the tumor itself. This is the strategy the tumor is utilizing to protect itself from the immune system. It's secreting cytokines and molecules into the microenvironment that make their way into the bloodstream, that activate and attract myeloid cells from the bone marrow, where they're normally made and normally have a very important physiologic function in our body. They hijack that mechanism and attract the myeloid cells into the microenvironment of the tumor. And once they're programmed by these molecules and cytokines secreted by the glioblastoma tumor cells to become a protective army against immune effector cells, our body's defenses trying to attack the tumor. So this is what research has identified as a very critical component of the strategy glioblastoma tumors are utilizing to protect themselves from the immune system. And if we can even partially diminish that or alter that, I think that will be the key factor to allow immunotherapy to have an impact with this disease. I think until we do that successfully, somehow figure out how to overcome this army of suppressive myeloid cells the tumor is exploiting, immunotherapy will not work in this disease. That's going to be the critical step we'll need to overcome. And it may only need to be done to a partial degree. It may not need to be done 100%. If we could take care of or flip 20 to 30% of these cells, that may be enough to allow the immune system to be able to have more of a chance. So I think being able to demonstrate that a therapeutic, for example, what we're talking about today, Temphron, which is specifically designed to have an impact against this biologic aspect of the tumor, if we can demonstrate in the tumor microenvironment that it indeed is having an effect in changing some of those cells to not be suppressive, but to function in their normal capacity, which would actually help the immune system, that would be a tremendous step forward in our efforts to try to have immunotherapy have an impact for this disease. We need to demonstrate, of course, along the way that this treatment is safe. Patients don't have significant side effects from the therapy. We preserve quality of life and neurologic function, and also that we prevent we prevent tumor from growing and progressing. Glioblastoma, by definition, is an infiltrative disease. The cells migrate outward into the brain, and as they do that, they can be highly destructive. So if we can stop progression of the tumor, that itself is a major advance, because we're limiting the ability of the tumor to grow and cause more damage and harm to patients. Ultimately, we want to have our patients live longer and certainly ultimately be cured. But at least these steps along the way, I think, would be very, very exciting if we were able to demonstrate that.
And is there a particular subset of patients that you think might be good candidates for something like this, or a certain stage of disease? With that immunosuppressive profile of the disease, some very late stage patients, unfortunately, are in a very weakened immune state. What is your opinion on that?
I have to agree, Brad. I think the optimal scenario to evaluate an particularly an immune-based treatment, one designed to help our immune system successfully attack the tumor, should be early in the course of the disease. I would argue in newly diagnosed patients, once they've gone through that standard of care that I described at the beginning of the chat, with chemotherapy, radiation, their immune system is weakened and continues to weaken over time. And that, even for an immunotherapy that can be successful, will make it much more difficult, because the immune system is eroded over time. Its ability to respond is diminished because of the effect of chemotherapy and the debility of the disease that ensues. So I think moving upfront into newly diagnosed patients makes the most sense to start with. The other important factor for our patients, I think, is to try to limit patients who are needing particularly high doses of corticosteroids. We use steroids. Unfortunately, they're a part of our treatment that has been required historically, because the tumors cause a lot of inflammation and swelling in the brain, and that feature can cause patients to have neurologic deficits, bad headaches, seizures. So we've used highly potent steroids like dexamethasone or decadron to decrease inflammation associated with the tumor. But that can be detrimental to immunotherapy treatments. So I would argue newly diagnosed patients where there'd be a stronger immune system and less of a likelihood of the need or requirement for dexamethasone would be the best scenario to evaluate an immunotherapy-based treatment.
Okay, and as we're a little bit past the bottom of the hour, I want to remind our listeners, if anything that's being discussed today raises a follow-up question in your mind, there's a Q&A function, so please ask, and we'll try to get to those later on in the presentation. So now I'm going to go back to you, Dr. Naldini. We just heard from a clinician what he would like to see from a treatment like this. Wondering what the initial clinical data is showing so far. For example, do the cells that you're making persist in the body? Are you seeing rapid engraftment of the engineered cells and the hematopoietic reconstitution after treatment? Have there been any systemic or localized toxicity or tolerability issues? And have you seen evidence that the cells you're engineering are making it to the tumor, like Dr. Reardon said he's interested in seeing?
Yes, of course, these are the key questions you wanted to address in the trial. So although all phase 1-2 trial are, of course, aiming initially at safety, There's much more than we need to show than just being safe in this type of treatment. First of all, it does look to be safe. We have, in a typical dose escalating scenario, we have been able to move up with the dosing, moving from a cohort of 3 patients into the next one, and without any dose-limiting toxicity emerging so far. But most importantly are the questions that you mentioned. So what about the engineered cell? Are there, and do they do their business? And we have a very good sense of that because we are used to monitor engineered hematopoiesis in our clinical experience. And the cells are there. They are engrafting into the patient without any toxic or adverse effect on the recovery of hematopoiesis after the process of autologous transplantation. The cells are found in terms of genetic marking into the circulation, to, at least initially to the level that we put them in. So they reflect exactly the dosing. They tend to lower down this concentration with time and then plateau and stay longer until the latest follow-up. So they are there. And importantly, there's no sign of peripheral exposure to interferon. So the regulation that we have built into the vector is working suitably. Patients have a significant fraction of engineered cell in the blood and presumably moving also into the tissue, and they don't see, there's no sign of exposure to interferon. There is a very minimal level of interferon in the blood that we can find, which was not present in the control or before the therapy. So telling us that the gene is there, but is not on, except hopefully. So what about the tumor then? That's the key question. Of course, it's more difficult to address this question because the tumor is inside the brain, and the only way we can really look at the tumor is if the patient do show progression and they are advised to undergo a second surgery for removal of the progressing lesion. This happened, and remember, these are patients who are all expected to progress, and in particular, as we started the trial, also the dosing regimen are on the low side. So we did expect progression at a certain point, and we could sample some of the progressing lesion. And although these remain anecdotal because there are few actual numbers are few, we could document the presence of the modified cell within the tumor, and also sign of interferon activation at the tumor site. And in a particular, at least one case in which we could see a lesion that was did halt its growth after the treatment, but then surgery was recommended anyway for a number of other reasons. In that lesion, we found significant interferon response and also some indication of that reprogramming that we saw in the preclinical model, and also Dr. Reardon mentioned. So we do have indication of activity as well at the tumor site, and that's where we are now.
And you touched on persistence. I'm wondering if you can expand on that a little bit and talk about how important you believe persistence of the cells is. And, when I think of gene therapies in the rare diseases context, I think of one and done, that, maybe a lifetime treatment. I'm wondering what's your expectation in this case? Do you expect that these cells might stay with the patients for life, for example?
Well, that was part of the rationale. I mean, at least for prolonged persistence of the cell, of the engineered cell. That's why we do start from hematopoietic stem and progenitors. Monocytes are short-lived, even within the tumor. And so if we were just to deliver monocyte, that would be a very short-lived treatment. We also learned from CAR T cell that although the T cell may be longer living, they often are not enough, sustained in the body patient for sustained delivery. Cancer need sustained delivery of biotherapeutic because the cancer is before really halting grow, unless it's full eradication, the cancer will develop resistance and evasion. So you need to have the capacity for continued delivery and to maintain that activity. So we do see persistence of the cell. They may not persist for life. In our preclinical model, there is a tendency in the longer term eventually to exhaust, but that's over probably several months, if not a couple of years. Because we don't see side effect, this is at the moment something we are comfortable with. In the laboratory, we have built a second generation strategy, which will also allow to turn off potentially the vector in case we have full responses, but that's something to come.
Okay. And I'm actually I see Q&A, somebody brought up a good question that I want to make sure that we highlight right now. So this person, it's Ren Benjamin from JMP Securities. He's asking, what specific monocyte cell is this and how do you isolate it? We're talking about Tie2 monocytes, correct?
Yeah, so again, let me clarify. What we isolate and modify are CD34 positive cells. So these are the progenitors, which are universal progenitors, and which will give rise to all type of hematopoietic progeny. The vector will be within all of this cell, but will only turn on into a specific subset of monocyte, which are the so-called Tie2 positive monocyte, which is a specific phenotype typically found and enriched in tumors.
Okay. And you heard from Dr. Reardon about what type of patients, earlier stage patients might be the best responders to this, but I'm wondering in your preclinical work and looking in the science and really even your early clinical data, if you're seeing any biomarker strategy, for example, that may help you do patient selection, and if you have any thoughts about that.
Well, we obviously we are best preferably targeting tumor in which there is a substantial recruitment of these monocytes, and we know glioblastoma is one of those, and most of glioblastoma have that infiltration. We know that an interferon gene activation signature is associated with better prognostic outcome in many tumors, including this glioblastoma, of course. And in terms of the mechanism of action of this of interferon, we know there is a general suppression of growth, most likely through suppression of angiogenesis at the tumor site, concomitantly with activation of the immune response, which will build a tumor-specific protective immunity. Now, in principle, and I think a very relevant point that was raised by Dr. Reardon is to enable this immune response build-up to really reach the best of our therapeutic strategy. And that means to have a non-compromised immune system and potentially avoid immunosuppressive drugs. So these are some of the strategy that we should like to really focus on. We don't really have a clear this identified biomarker at this point. This is something we are still working on, and it's too early on the trial to really comment on this.
Okay. And Dr. Reardon, let's go back to you. In Cancer Today, we talk about combination therapies for all different types of cancers. What are some of the things that you think might make sense to combine with this, and why?
I think this treatment lends itself very well to combinatorial therapy, which I think for this disease is a requisite. We're not going to be able to have a therapy that works as a single agent and cures these patients. These tumors are too complex and have significant heterogeneity, other multiple mechanisms of giving the cells growth advantage that combination therapy, whether it from an immunotherapy or chemotherapy, any approach is going to require a combination approach. But this approach, I think, targeting the microenvironment and the critical factor in the microenvironment that the tumor is exploiting to protect itself, these myeloid cells, would be it would be very exciting to combine it with strategies that could increase immune cell activation and movement into the tumor. For example, radiation therapy, which is a routine standard of care, as I mentioned, for patients, kills tumor cells. The tumor responds temporarily. When the cells die, they release tumor antigens that can trigger an immune response against the tumor. That immune response today is effectively blocked and negated by the myeloid cells in the microenvironment, so that it can't amount to very much. But if we could blunt that myeloid suppressive effect, those T cells that are activated by immunogenic cell death induced by radiation therapy, for example, may be able to come in and have a meaningful benefit. So right off the bat, even with a standard of care therapy, there would be a good rationale for combination strategies. But we know there are other protective immunosuppressive mechanisms in the microenvironment of these tumors, upregulation of immunosuppressive modulating molecules like PD-1, PD-L1 in the tumor microenvironment, and other inhibitory molecules. So combinations with some of the checkpoint inhibitory immune checkpoint therapies would make good sense. Strategies that could also enhance immune sensitization, robust vaccine strategies, and even in situ local therapies. There's a lot of interest now in oncolytic viruses in glioblastoma being injected into the tumor, where they can generate a very potent immune response. We refer to it as an in situ vaccine that could partner in a very complementary way with a strategy targeting the myeloid component of the tumor. So there are a very broad, I think, list of exciting possibilities that could be readily combined with such a strategy that Temphron is based on.
Well, thank you. Dr. Naldini, one last question for me, and then we'll go to our analyst questions. We've talked all about glioblastoma today, but one of the things that excites me about Genenta is that Temphron is tumor and antigen agnostic. I'm wondering, what evidence do you have of potential synergies for Temphron in combination with other immuno-oncology products? And what other solid tumor cancer types do you foresee applying an approach like this?
No, thanks. Let me start from actually following up on the specific question on glioblastoma and also synergistic treatment that Dr. Reardon mentioned. I couldn't agree more with what we said. We modeled that in preclinical studies. We of course saw that in glioblastoma model, our treatment delays the glioblastoma growth in pretty all the mice treated, but some of the mice did in fact fully clear the tumor, but just a fraction of them. And so we have been working also to look at combinatorial treatment, and in particular immune checkpoint inhibitors and CAR T cell have both been very synergistic to our strategy, which makes sense because in principle we activate the immune system in terms of unleashing the capacity to present antigen from the tumor through this remodeling of the microenvironment. And then if we can empower the T cell response either by checkpoint blockade or by even providing a CAR T, which could trigger cell death to begin with the spreading, that's very powerful combinatorial approach. And going to your specific point, all of what we have been discussing in principle may apply, as we said, to other tumors. So we are agnostic, as you said, to the tumor, and in particular to the tumor antigen. by unleashing the immune response, we let the tumor present the antigen against which the immune system can respond. We know this is probably true for most tumors. Some tumors have probably more than other. Most of these may be private antigens, so they are specific even for an individual tumor. So that's the benefit of our strategy. We don't have to rely on a universal tumor-specific antigen against which a CAR-T would have to be delivered, and we don't fear too much the escape from a single, immuno-targeted strategy. As long as the microenvironment allows the deployment of an immune response in terms of presentation of antigen and effective effector cell activity, this can work. We have modeled this in other solid tumors. There's a number of them which also would fit very well this strategy. We are considering broadening we have been talking about moving into liver cancer, both of different type and origin, and as well as breast cancer. There is preclinical data for that. And I think the key principle of immunoactivation by microenvironment reprogramming is broadly applicable to solid tumor.
Great, thank you, Luigi. At this time, we will be conducting a question and answer session. As a reminder, if you would like to submit a question, you may do so by using the Q&A text box at the bottom of the webcast player or by emailing your questions to questions@lifesciadvisors.com. Please hold for a brief moment while we pull for questions. So our first question comes from Tony Butler from Roth Capital. Tony, you may go ahead and unmute your line.
Yes, sorry, Tara, I had a little trouble. Thank you. Thank you, Dr. Naldini, Dr. Reardon, appreciate it. Brad, Carlo, good morning. My question is directed toward you, Dr. Reardon, and it's really three parts. Number one is if Temphron's given, Dr. Naldini alluded to the notion that there would be suppression theory, suppression of angiogenesis. So do you think that perhaps bevacizumab might be of use at the same time that Temphron might be given? That's question one, if I may. And question two is, in the combination discussion that you alluded to with checkpoints in particular, I'm curious though, if you were to deliver anti-PD-1, for example, in conjunction with Temphron, does it not inherently cause inflammation, perhaps driven by gamma interferon, and therefore the necessary need for steroids? That's aside from the notion that the patient has still residual glioblastoma cells and has had radiation and some chemotherapy, all delivering some inflammation. And I guess the notion is, I don't know how you get around corticosteroids. And I'm curious more, I'd be interested in your discussion in greater detail around how you might think about that from a clinical design standpoint. Thank you very much.
Those are really very insightful questions, Tony. The first one regarding a possibility of combination with bevacizumab, given that the Tie2 macrophages that are targeted with this therapy, we know are critically related to the angiogenesis, the ability of these tumors to generate a remarkable increase in blood supply, blood flow, blood vessels to feed the tumor. That's one of the hallmarks of glioblastoma, and being able to block that angiogenic component has been a therapeutic strategy that's been evaluated in many ways for this disease. No question, glioblastoma is amongst the most angiogenic of all cancers, and the type 2 macrophages are an important component. So this therapy has I think an additional benefit, if you will, of by blocking those cells, may also have an effect against the advantage the tumor utilizes with its angiogenic capability. Bevacizumab in combination is an interesting strategy or potential. I think we didn't talk about that, but in the US, bevacizumab is approved for the treatment of recurrent glioblastoma. So it could be applied in combination therapy, and it may have some complementary benefit. I think it would be very intriguing to see. It does tie in with your second question, which is the question about the inflammation in the tumor, the need for steroids that I mentioned because of the inflammatory reaction associated with the tumor. We've relied on highly potent steroid medications like dexamethasone to counteract that and improve neurologic deficits so patients' quality of life is not diminished. But we have used in many of our immunotherapy trials now going forward low doses of bevacizumab. It's not anti-inflammatory, but it does have an anti-permeability effect that can accomplish the same thing that steroids do, but by a different strategy. It blocks or blunts the leakiness of the blood vessels in these tumors and the associated swelling and edema without compromising the immune system. There's not an anti-inflammatory component. So we need better strategies to block the edema and swelling that's inherent with this tumor as it's infiltrating into the brain. But at least one strategy we can use that's had some benefit is low doses of anti-angiogenic agents like bevacizumab. And that will hopefully allow us to avoid the need for steroids. The combination of immunotherapy treatments can, you're correct, increase the inflammation that's already inherently present in the tumor. So we do have to be careful about that and think carefully about dosing schedules and strategies to be able to try to avoid that in a way that would cause more difficulty for patients. We try to walk a fine line, and you're absolutely right. It is a challenge.
Thank you, sir.
Great, thank you for the questions, Tony. Our next question comes from Noreen Quibria from Maxim Group. Noreen, you may go ahead and unmute your line.
Oh, hi, good morning. Sorry, I was having trouble unmuting. Thank you for this. This was a great overview. Sort of piggybacking on what Tony asked, I'm curious, if a patient has prior bevacizumab experience, does that prior Bev experience have any impact on the new agent or therapeutic that's being tested in GBM? This is for Dr. Reardon.
It really depends, Noreen. I don't think it's necessarily the case one way or the other. It depends on how much exposure for how long, and if the tumor has developed a resistance and progression. So bevacizumab and anti-angiogenic agents can have an improvement for patients. And I think it's primarily related to their anti-angiogenic effect and their anti-permeability effect, where patients can have a window of time where they're benefiting. But at some point, the tumor develops strategies to overcome that blockade of the vascular endothelial growth factor that bevacizumab and other therapeutics are targeting, and becomes resistant. And once it becomes resistant to bevacizumab, we haven't come up with any therapy that's worked with any consistency for patients. I don't know in that context if trying to have an impact on the myeloid component of the tumor or the Tie2 targeting macrophages may benefit theoretically, it's possible it might, because those macrophages are critically integrated in the angiogenic effect of the tumor. But I think it would be, in general, in our experience, that's a very challenging subgroup of patients to have to treat. So if patients are exposed to bevacizumab, my recommendation or preference would be to try to use this therapy before, either have the patients off the bevacizumab or use it before they've developed resistance while it's still helping them.
Okay, got it. That's really helpful. And Dr. Reardon, just broadly speaking, what are some key studies in GBM that you're watching in the space right now?
Well, one of the things that we kind of stumbled on accidentally, it's so interesting how much sometimes serendipity plays a role in how things evolve in our fields. But when the initial studies of checkpoint blockade, the phase 3 studies, moved very quickly out and testing these agents for glioblastoma. And unfortunately, as I mentioned, they were all negative, didn't show a survival benefit with single agent PD-1 blockade, for example. So we designed some studies looking at neoadjuvant dosing, where the checkpoint blockade was given for 1 or 2 doses prior to scheduled surgery for patients. And the idea was to, once the tumor was resected, to understand if the drug was doing anything. Was it modulating the immune microenvironment? Was it having any intended effect? Because we wanted to understand why the phase 3 studies were negative. And we kind of hypothesized these drugs really didn't do much in the tumor microenvironment of GBM, and that's why the phase 3 studies were negative. So we designed neoadjuvant studies of checkpoint blockade prior to surgery for recurrent GBM patients. And they did confirm that we did see a flip or transformation of immune activation, gene expression profiles. We did see an influx in immune effector cells into the microenvironment. We saw an expansion of T cell repertoires peripherally. So the studies did show that these drugs do have a positive effect of activating the immune system. So it didn't give us an answer why the phase 3 studies were negative, but at least it provided some proof the drugs do have some beneficial effect. What we completely didn't expect in this particular initial 32 patient randomized study of neoadjuvant checkpoint blockade was a nearly 2-fold improvement in survival for the patients who got a dose of neoadjuvant PD-1 blockade compared to the 16 patients who didn't get a dose beforehand, and everybody got checkpoint blockade afterward. So one of the really exciting, I mean, that was very exciting, and there have been, as you may know, other studies in other cancers showing neoadjuvant dosing of immune checkpoint therapies having an interesting therapeutic efficacy benefit. So we're now trying to better understand that, and there are a number of studies in GBM now looking at neoadjuvant dosing of some of these immunotherapy approaches, and we hope that those initial remarkable survival benefits, what I've dreamed about for my whole career, to see those significant improvements in survival, that we can validate and reproduce that with the ongoing studies that are being done. I think the oncolytic virus work that's ongoing in a subset of patients, there's several of those that look very exciting and promising. And I think they're going to need to be combined with treatments to get to their full effect. But I think those are 2 examples where we've seen there's reason to be hopeful and excited about the future and follow-up studies.
That's great. I apologize. One more question. Actually, this is for Professor Naldini. You sort of touched on it before. Can you sort of discuss why interferon alpha as the payload in Temphron versus something else?
Well, we of course, this is a platform for delivery of different cytokines. We have been studying a few of them, many of which have the potential, of course, to activate immunity. Interferon alpha has been the most effective in terms of both benefit and lack of adverse effect. Let's say now in terms of ease of administration today. So that's why we have been we continue to work on that. We have evidence that other cytokine like interleukin-12, interferon gamma, for instance, may also be administered to provide anti-tumor effect, and we are studying also now combinatorial potential delivery. But interferon alpha has been the leading candidate until now for the probably pleiotropic activity, both on the innate and adaptive immune effectors.
Great, thank you. That's it for me, thanks.
Thanks for the questions, Noreen. We have time for questions from 2 more analysts, and then we will be wrapping the meeting up with closing remarks from Chief Medical Officer Carlo Russo. So our next question comes from Justin Zelen from BTIG. Justin, please go ahead.
Thanks for taking the question. So Dr. Reardon, given how aggressive GBM is and the limited durable treatment options out there for patients, I'm curious what you'd consider to be clinically significant for a new therapy in terms of clinical outcomes.
Justin, I think it depends. I think you're alluding to efficacy endpoints, which obviously are the bottom line. We want to improve survival for our patients. Unfortunately, the bar is set pretty low in this disease. So if we saw a drug showing a say 25 to 30% improvement over the historical standard of care in either progression-free or overall survival, I think that would be, I know it doesn't sound very exciting to many of you, but in our field, dealing with such a challenging disease, 25 to 30% improvement, which amounts to a few months, basically. It's not very long, but that would be an exciting signal for us to want to move forward and test further. Is that what you were looking?
Great, thanks for taking it. Yes, that's very helpful. Thank you so much, and thanks for taking the question.
Thanks for the questions, Justin. Our next question comes from Emanuela Branchetti from H.C. Wainwright. Emanuela, please go ahead.
Good morning, everyone, and good afternoon, Professor Naldini. Thank you for taking my questions. I guess for both Dr. Reardon and Dr. Naldini, what can you tell us about the expected kinetic of responses based on Temphron mechanism of action? Would you expect to see pseudoprogression perhaps before seeing actual responses in patients?
That's a great question, Emanuela. I wouldn't be surprised if we do. We know if the immune system is able to break down some of the barriers the tumor has put in place and can make its way into the tumor and activate, that we might expect to see some inflammatory pseudoprogressive changes on imaging. I would actually find that pretty encouraging if we saw that as a validation that the drug is modulating the microenvironment. What we typically would expect clinically, though, of course, is things to stabilize and then over the next several months, maybe gradually improve as the immune system is doing its job. But I wouldn't be surprised if we saw that kind of a evolution of change with a drug that's working via such a mechanism.
Yeah, I can second, and I agree with this answer, Manuela. I think we need some times for because we administer progenitors, we need some times for the reconstitution of the lineages by the engineered cell. So there is a time gap in which we expect to have before the therapy, let's say, can establish in terms of delivery. At the same time, a component of the therapy, of course, is recruitment of the cell at the tumor site, depending on the design of the trial. In the case of glioblastoma, this is after surgery, the primary surgery. So it may also require that there is some initial regrowth of the lesion to recruit the monocyte, which then will deliver the activity. So it's very possible that we see this sort of initial pseudo progression and response and so on. That's something we are carefully looking at in our patient now.
Great, thank you very much for that. And perhaps this is for Professor Naldini. You discussed potential target indications beyond GBM. Looks there are, different potential options, but I'm curious to know how are the data in GBM expected to guide your decision on the next indication? I guess what are the data that could help guide your decision and what we should keep an eye on?
Well, obviously the validation of the platform in terms of establishing the engineered hematopoiesis and the capacity for delivering biological activity related to interferon signal in the tumor. We expect to be able to establish that by a collection of data from analysis of tumor specimen, as I mentioned, from progressing lesion, imaging studies, and also monitoring the immune system evolution, both the tumor but as well in the periphery. We are looking at clonal changes in periphery as well. So the key, I think, in terms of platform validation would come, of course, any additional benefit in terms of therapeutic benefit in terms of PFS and so on would be as well crucial.
Got it. Thank you for that. And maybe related to this question, could you describe the current process and time between apheresis and reinfusion and how these could impact the application to other solid tumors beyond GBM?
So the processing of the cell is fast, is over 24 hours, essentially, after collection. And currently, there is a significant delay in terms of release, which is true for all cell therapy products. Essentially, once they are manufactured, they are frozen, and the time to release may change from 2 to 4 weeks. Currently, is at least 4 weeks. There's way to shorten that. This has not been an issue into this trial because, of course, after the cell harvest is done, of course, after the patient are recruited, after the first surgery and full diagnosis, but then the patient undergo harvest of the stem cell, and then they'll do the routine radio and chemotherapy course. And so during that time, there is plenty of time to manufacture the product. In other setting, of course, we could maneuver this timing. And as I said, manufacturing can be, for this product, is very fast. We also add strategy for faster release in some trials, including fresh cell release in some disease in which we needed that. So I think there is quite a room for fitting this strategy of manufacturing to the cancer type we will be treating.
Great, thank you very much.
Thank you for the questions, Emanuela. I'll now turn the call over to Carlo Russo for closing remarks.
Thank you very much, Tara. It's been really a privilege for Genenta to be able to host this discussion. I want to thank Professor Naldini and Professor Reardon for the very elegant and thoughtful discussion on the unmet medical need of this population of patients that are really suffering for a terrible disease, and the potential of a technology that Professor Naldini and Genenta are developing to address the unmet medical need. I want to just add that your question regarding the pseudoprogression is a very important one, and we have a presentation at ASCO in June, as well as next week in DC, on preliminary data that were discussed today in this Fireside chat. So I encourage you to go and look at the presentations of those meetings. With that, I found the discussion very informative. I think it is the type of focus that really we should put on the patient need and how the patient could be addressed. And I'm very thankful for the patient population that is being willing to participate in our study. We need to keep the focus on the patient needs and what we're trying to do. I also want to thank Brad. It's been a professional way to moderate the discussion. And thank you, Brad, for the work you've done. And also want to thank LSI Tara for organizing this fire chat. I trust that everyone attending this meeting found it quite informative. So thank you, everyone.